Okay, so today I'm going to be discussing what we did in the lab this week in our respiration lab, and I'm just going to basically go over the experiments. Respiration is basically when organisms take their food and they break it down and they create usable energy from it. The usable energy they make is in the form of adenosine triphosphate, or ATP. It's the energy current to the cells, the way they transport and use energy. And our first experiment was looking at aerobic cellular respiration. I'm not going to go over the entire process of aerobic cellular respiration. You should read your lab manual about that. But basically, the idea is that in aerobic cellular respiration, the organisms will take glucose, combine it with oxygen, and they will create carbon dioxide, water, and ATP from that. So we have carbon dioxide gas given off as a waste product of aerobic cellular respiration. And we're going to use that fact to do our experiments today. Our experimental organism is yeast. Yeast is, of course, a fungi, and it can do both aerobic cellular respiration and also alcoholic fermentation. That's what makes it valuable for our experiment today. And for our first experiment, it was exercise 8, demonstrating the effect of substrate type on carbon dioxide production by yeast. Basically, the idea is that there are certain kinds of sugars the yeast can use to do aerobic cellular respiration, certain kinds it can't. And our experiment found out some of the ones it can use and some of the ones it can't. So we had different glass test tubes. We had some glass test tubes of water for our controls, and we had sugars like glucose and fructose and galactose, and sucrose, and maltose, and also lactose. So these are all kinds of sugars, with the exception of our controls, the water, and we wanted to see what kind of results we would get if we added the yeast. Could the yeast digest these sugars, or could they not? Organisms that can't digest those sugars can't digest them because they're lacking the correct enzymes. You can imagine if you went out in the woods and you picked up a stick and started eating it, you wouldn't be able to get energy from it because you're lacking the correct enzymes to digest that cellulose sugar. But something like a termite could, because it does have the correct enzymes in the bacteria in its gut. So let's look at the results we got for our experiments. And the idea is that we have, in each of these tubes, we have a glass tube of the sugar along with some yeast, and it's suspended in the tube of bromothal blue. Our bromothal blue at neutral pH is green. When it becomes acidic, it turns yellow in color. Now, what makes it acidic if there's aerobic cellular respiration is the fact that carbon dioxide gas is released. When carbon dioxide gas is released inside this tube, it will dissolve inside the bromothal blue. When it dissolves inside the bromothal blue, it forms carbonic acid, and it lowers the pH, and therefore it makes the bromothal blue change color and become yellow. So if you see a yellow color in the tube, it means there is aerobic cellular respiration occurring. If it stays green, it means there's not. So, for example, our two control tubes with the water did not have any color change, they just stayed green. Our tube with the glucose did turn a little bit yellow, and you can see some foam, that is carbon dioxide gas appearing, because there was respiration. You can see we had, and let's see, tube 4, it was fructose. You can see there's a lot of yellow color and lots of foam. That means the fructose did react with the yeast. And tube 5 was galactose, and nothing happened because it can't digest galactose. Tube 6, sucrose. It can digest sucrose. You can see it's got some foam and you see yellow color. Tube 7, maltose. Still got that yellow color, which means that there was aerobic cellular respiration. Also, you can see some carbon dioxide bubbles. And 8 was lactose. Yeast cannot digest lactose, so there was nothing occurring. Okay, and so that was our first experiment. Our second experiment was alcoholic fermentation with yeast. There's two kinds of fermentation I want you to know about: lactic acid fermentation and alcoholic fermentation. You should read about them both in your manual. But here's what our experiment was. These are fermentation flasks, and we had water in this one, we had glucose in this one, and we added yeast cells to each of them. Now, they're called fermentation flasks because they work well with fermentation. Basically, yeast can do both alcoholic fermentation or aerobic cellular respiration. It would prefer to do aerobic respiration because it produces a lot more ATP. However, if it can't do aerobic cellular respiration because it's lacking oxygen, it'll switch over and do alcoholic fermentation. It doesn't get a little bit of ATP, and a little bit is better than nothing. So all these yeast cells here were underwater, so they didn't get much oxygen. So because they couldn't get oxygen, they didn't do aerobic cellular respiration. So they switched over and started doing alcoholic fermentation. When they did alcoholic fermentation, they produced carbon dioxide gas as a byproduct, and the bubbles get trapped in the tube, and we were able to measure how many milliliters of gas was produced in the tube. This one produced, looks like, about 2 1/4 milliliters of carbon dioxide gas. So that's a demonstration of fermentation by yeast. And you should know all about lactic acid fermentation and alcoholic fermentation from reading your manual. Now let's see, our next experiment was demonstrating electron transport in yeast using a dye indicator. It's over here on the monitor. Okay, we got living yeast cells, and you can see something very interesting. We have dyed them with methylene blue, but not all the cells are blue. Some of them are blue, some of them are white. Okay? They're not all dyed the same color blue because they're doing different things. And methylene blue is a special chemical called a redox indicator. It means it changes color depending on whether there's oxidation or reduction reactions occurring. And methylene blue is blue when it's oxidized. That means when it has fewer electrons, and it turns colorless when it gains electrons. You can remember reduction is gain and oxidation is loss. I remember the phrase oil rig. So oxidation is loss of electrons, and reduction is gain of electrons. And the organisms, the yeast cells that stay blue in here, are blue because they are not doing aerobic cellular respiration, they're not doing fermentation, and so the methylene blue is not becoming colorless. The yeast cells that are colorless or white are that color because they're having aerobic cellular respiration or alcoholic fermentation occurring, and that's changing the color of the yeast cells. And your section of your lab manual, exercise C on page 79, is actually incorrect. There's a handout you can download from Blackboard that has a correct version of exercise C. So you should study the handout from Blackboard and not what's on pages 79 and 80. Okay, let's look at our next experiment. It was, let's see, exercise D, demonstrating the effect of catalase in aerobic cellular respiration. And basically the idea is that hydrogen peroxide is a toxic byproduct of aerobic cellular respiration. So people use it as an antiseptic because it kills living cells, but of course it will kill your body cells as well as bacteria. So since it's a byproduct of aerobic cellular respiration, all organisms that use aerobic cellular respiration have to have a method of getting rid of hydrogen peroxide. And many organisms do that using catalase. Catalase is an enzyme. Remember, enzymes speed up chemical reactions. And specifically, what catalase does is it speeds up the breakdown of hydrogen peroxide into water and oxygen. So I've got a demo here to show you. We have some Bacillus megaterium bacteria. If you add hydrogen peroxide, we'll be able to see if it has catalase. Remember, there's a reaction occurring in the tube if catalase is present. You can see there's bubbles occurring in the tube. So the catalase in the Bacillus megaterium is breaking down the hydrogen peroxide into oxygen bubbles and also water. So this proves that Bacillus megaterium has catalase. And remember, all organisms that use aerobic cellular respiration have to be able to get rid of hydrogen peroxide. And one of the ways that they do it is catalase. So that means that Bacillus megaterium probably does aerobic cellular respiration. And we know that because it's catalase. There are some organisms that do anaerobic respiration that have catalase, but they're not particularly common. Okay, our last experiment is demonstrating carbon dioxide release during respiration. It's a very simple experiment. So we've got different tubes with different specimens in them, and they're suspended above Bronthal blue. Remember, Bronthal blue will turn orange when there's carbon dioxide production in the tube. And so we can see some of them turn green, stayed green, some of them turned orange. So all the ones that had aerobic cellular respiration occur turned orange. So our control stayed green. Our germinating pea seeds turned orange because they respired. The boiled plant seeds stayed green because there was no respiration. And the small plant, living small plant, turned orange because there was respiration. The leaf turned orange because they had respiration. So the insects and the rocks, of course, did nothing because they were just a control. So that's basically a summary of experiments in this lab. You need to read over basically this step: alcoholic fermentation, lactic acid fermentation, aerobic cellular respiration, and anaerobic respiration.